Hello students, welcome to EPG Pathshala. I am Dr. Lokesh Chandra Mishra from Hansraj College, University of Delhi. Today we are going to talk on module Giardia mode of infection from paper Biology of Parasitism. The objective: the module has been designed to make you understand general aspect of Giardia infections, mode of infection and epidemiology, diagnostic tools, Giardia symptoms, prevention, treatment, and finally virulence. Introduction: Giardia is a microscopic parasite is responsible for disease giardiasis. There are many species of Giardia: Giardia duodenalis, which is synonymous to Giardia lamblia, Giardia intestinalis, can infect human beings. Giardia rd, Giardia psittaci can be found from birds. G. agilis can be found from amphibians. G. muris and G. micropti can be found from rodents. Giardia is found in soil, contaminated food, water which can be drinking or recreational water is the most common mode of transmission. Giardia can survive outside the body for long period of time due to the presence of an outer shell which is a covering. Giardiasis is the most frequently diagnosed intestinal parasitic disease in various countries. Symptoms may vary from no symptom to diarrhea, gas, greasy stool, abdominal cramps, nausea, vomiting, upset stomach, and dehydration. Severe giardiasis can cause delay in physical and mental growth with slow development and severe malnutrition in children. Persistent infections can cause weight loss and malfunction in fat absorption, lactose, vitamin A, and vitamin B12 in adult as well. Giardia duodenalis has an invariant morphology, host specificity, and transmission pattern which requires complete genetic analysis of cyst and trophozoites. Complex epidemiology of infection can be studied with the help of latest tools and techniques. Diagnosis can be done by demonstration of trophozoite in direct fecal smears endoscopy to demonstrate the organism in duodenal aspirate and enzyme-linked immunosorbent assay, which is also called as ELISA, for Giardia antigen in feces. This is based on antigen and antibody interaction. Giardiasis treatment include antimicrobial drugs such as metronidazole, nitoxonaride, good for children, tinidazole, paroxymycin, quinacrine, and furosemide. Infection can be defined as the invasion of and multiplication of microorganisms such as bacteria, viruses, and parasites. An infection can cause no symptom and be subclinical, or it may cause symptom and be clinical apparent. An infection may stay localized or it may spread systematically through the blood or lymphatic vessels. Microorganisms like few bacteria type that subsist naturally in the body are not considered infections. Infection and epidemiology. An organism living in or on another organism and deriving nutrients to survive is called as parasite. Parasite flourishes at the cost of host animal. Giardiasis is a diarrheal disease caused by the microscopic parasite Giardia. Once a person or animal, for example, cats, dogs, cattle, deer, etc., has been infected with Giardia, the parasite lives in the intestine and passes out with fecal matter. Giardia can survive for weeks or months outside the body. Now, mode of infection. Giardia duodenalis, which is synonymous to Giardia lamblia or Giardia intestinalis, lives in the duodenum and jejunum. Giardia can cause infection through: number one, consuming water or ice from unhygienic water sources like water from lakes, streams, or wells. Number two, ingestion of Giardia accidentally from exterior, for example, bathroom handles, changing tables, diaper pails, toy, etc., that contain feces from an infected person or animal. Number three, swallowing water during water sports, especially in lagoons, river, canals, springs, lakes, and ponds. Number 4. Uncooked food containing giardia. Number 5. Close contact with somebody suffering from giardiasis. Number 6. Roving in countries having high giardiasis occurrence. The infection is never through blood contact. People get infected with giardia when they eat or swallow something which is contaminated with feces from infected humans or animals. In figure 1, we have shown interaction between the domains responsible for giardia infection, in which human beings, pet animals, wildlife, and livestock interact with each other, and with constant touch in environment they interact with each and every component of environment also. Apparently, pristine water sources can also cause infection because of human or animal fecal contamination. Organisms can survive in cold water and can well tolerate chlorination of water systems. Ultimate resort at this stage is filtration of water. Other common mode of infection is person to person in daycare centers or workplace. Potential zoonotic risks can be rendered by infected dogs and cats, which can clearly serve as a source for contamination of the environment with the organism. The same threat can be posed by cattle, goats, and pigs, although some isolates from these species represent livestock, restricted strain, and have not been recovered from humans. Epidemiology. A considerable amount of data has shown that Giardia duodenalis should be considered as a species complex that comprises at least 8 distinct genetic groups referred to as assemblage A to H. The molecular characterization has shown the existence of different genetic assemblages in the G. duodenalis, also called as G. intestinalis species complex. Assemblage A and B, which cause infection to human beings, can also cause infection to other mammals. Transmission frequencies, animal reservoirs, and association with specific symptoms for assemblage A and assemblage B are not very clear. Besides, the occurrence and clinical significance of mixed infections and assemblage A and B are also poorly understood. Assemblage A has a broad host range which includes human being, primates, livestock, pet, wildlife, etc. It was also the most widespread genotype in a study of caged and wild Australian marsupials. Assemblage A seems to be common in wild mammal species such as beaver, cervets, muskoxen, and marine animals. Assemblage B is the predominant isolate type together with assemblage A and has even broader range which include human beings, livestock, dog, beaver, horse, rat, and muskrat. It can infect many species such as cattle, sheep, horses, dogs, cat, rabbits, guinea pigs, chinchillas, wild and captive mammals, and marsupials. This assemblage type seems to be uncommon in most mammals. In many studies, it was detected in only 0 to 2% of the population, in contrast to few occasions where it was most common genotype in a population. Assemblage C and D can reside in dog, cat, and wild carnivores and other canid including foxes, wild dogs, coyotes, and wolves. Assemblage C and D seem to be well adapted to these species. Assemblage C and D have also been detected in seals and kangaroos. Rare infections were documented in other domesticated and wild species, including a goat and a few cats. Assemblage E is usually the predominant type in livestock. It has been reported from cattle, yaks, water buffalo, farmed bison, sheep, goats, alpacas, and pigs. Studies have shown the presence of assemblage E in wild ungulates and wild deer. Assemblage F has been detected mainly in cats and appears to be adapted to this species. Few instances of infections in other species like dogs, pigs, and rodents have also been reported. About assemblage G, it has been seen mainly in rodents, including rats. Assemblage H has been reported from seals and gulls. Assemblage A, sometimes assemblage B also, is common in non-human primates, marsupials, and marine animals, in contrast to pets and livestock. The role of wild animals in maintaining and transmitting G. duodenalis to people is uncertain. Beavers have been apprehensive in zoonotic transmission according to epidemiological investigations of waterborne outbreaks. Since the conclusion was based on incidental evidence like recuperation of giardiasis from beavers in areas having contaminated water, the probability of contamination by humans or other animals could not be ruled out. Now we will talk about zoonotic potential of G. duodenalis. G. duodenalis is found worldwide with higher rate of infections in tropical and subtropical countries. Humans serve as key reservoir for G. duodenalis infections in people. It would be premature to comment on the extent of zoonotic nature in infections. The studies have not yet answered us about the cross-transmission frequencies and risk factors for giardiasis from animal contact. Genetic analysis of isolates observed in human beings and other animals have thrown some light on it. The studies have suggested that the rate of infection of G. duodenalis is more in human beings than other animals. Assemblage A and B cause giardiasis in people and can be found in livestock also. Reports have suggested that assemblage A genotypes in G. duodenalis databases were potentially zoonotic and have recovered from other animals also. Approximately 4 to 7% of assemblage B isolate were found to be potentially zoonotic. Several recent reviews have suggested zoonotic transmission from domesticated animals, but of minor importance compared to person transmission. On the basis of evidences, we can conclude that assemblage A and assemblage B isolates of G. duodenalis in animals ought to be considered potentially zoonotic. On the other hand, assemblage A and B isolates in people may have potential to infect animals. Diagnostics techniques. There are several methods to diagnose the infections of Giardia in human beings and other animals. Few of the important methods are: number one, demonstration of trophozoites in fecal smears. Giardiasis can be diagnosed by direct observation of trophozoites or cysts in the fecal smear. Stained preparations preserved with polyvinyl alcohol or 10% formalin is better. Multiple smears over the time must be done because of the discontinuous nature of Giardia shedding. The flagellated trophozoites is 9 to 21 micrometer long by 5 to 15 micrometer wide with teardrop and plumating motility. The two nuclei at the anterior end are also visible. Number two, fecal flotation with the help of zinc sulfate solution for cysts. Cyst can be detected by flotation method as trophozoites gets lysed by the solution of zinc sulfate. D. duodenalis cysts are approximately 8 to 15 micrometer long and 7 to 10 micrometer wide with oval shape and 4 nuclei. Repeated sampling may be required at the initial stage. Cysts are more likely to be found if specimens from 3 different days are examined as shedding occurs intermittently. Number 3, collection of duodenal aspirate for the direct demonstration of the organism during endoscopy. I have already told you about the endoscopy where a tube is inserted through buccal chamber or the mouth cavity to the stomach to observe direct through camera fitted at the anterior face. Besides, in case of chronic giardiasis, if the repeated stool examination are negative, the intestinal contents can be examined directly for trophozoites. The technique, also called as string test, which is an entero test, is useful in which the patient swallows a gelatin capsule on a string which on retrieval can be examined for trophozoites. Enzyme-linked immunosorbent assay, immunochromatographic test, and immunofluorescence assay, which is also called as IFA assays for Giardia antigens in feces. These are based on the concept of antigen-antibody interactions. These are highly sensitive and specific in humans in comparison to other animals. Rapid tests like ELISA should not replace routine ova and parasite examination by microscopy, which can also diagnose other diseases. Next, polymerase chain reaction PCR based molecular techniques can be used to identify the subtypes of Giardia. Genetic characterization of isolates can be employed only in epidemiological studies and research at the assemblage level. Last, serology can be used in epidemiological explorations and other research. Giardia can be cultured in vitro for the research purposes. Disease management. Giardiasis, also known as beaver fever, can affect anyone. Prime reasons for getting infection include close contacts with the people having giardiasis, children in childcare centers or creches, especially diaper-aged children, consumption or use of untreated or improperly treated water, lack of good hygiene practices, exposure to human feces, etc. Symptoms of giardiasis normally begin 1 to 3 weeks after infection. Giardia infection can cause a variety of intestinal symptoms. Symptoms of giardiasis last for 2 to 6 weeks, with few exceptions where these can last longer. Symptoms include diarrhea, flatulence, floating slimy stool, abdominal cramps, upset stomach, nausea, and dehydration. Persistent infection may cause weight loss in severe cases. On the other hand, symptom-less infection has also been reported in many cases. After 2 weeks of incubation period, sufferer can have an acute gastroenteritis with diarrhea, rarely with fever. Advanced stage can render a chronic syndrome of malabsorptive diarrhea, weight loss, and abdominal pain. Stool may be watery initially, then turns intermittent semi-solid with foul odor. Other symptoms include malaise, nausea, bloating, flatulence, etc. Persistent diarrhea can continue for the foreseeable future if not treated. Giardia parasite can infect all people but prefer infants and pregnant women who experience severe dehydration from the diarrhea caused by giardiasis. Dehydration can be life-threatening for infants, so it is particularly important to rehydrate them through proper oral liquid supplements and medications. In many cases, children do not have diarrhea but do have other symptoms such as nausea or upset stomach, tiredness, weight loss, or a lack of hunger. In such cases, they should undergo treatment. Giardiasis can cause weight loss and failure of absorption of fat, lactose, vitamin A, and vitamin B12 in severe cases. Diarrhea is the result of loss of disaccharide-based enzyme. The organism can colonize large portions of the small intestinal tract. The host inflammatory or immune responses add to the pathology of Giardia infections. Drugs for treatment of giardiasis include metronidazole, quinacrine, nitroimidazole compounds including tinidazole, ornidazole, etc. have also been found effective. Metronidazole, which has a brand name of Flagyl, is given in doses of 250 mg 3 times a day for 7 days for adults and 5 mg per kg 3 times a day for 7 days for children. Metronidazole is less effective than quinacrine to some extent, but is usually better tolerated. It has various side effects like metallic taste, dark urine or yellow urine, headache, and occasional gastrointestinal symptoms. Other less common side effects include nausea, vomiting, diarrhea, abdominal cramps, fever, and skin rashes. Quinacrine exhibit toxic psychosis with either depression or excitation, yellowing of the skin, and sclerosis. Furazolidone or furoxone, due to its liquid form, is suitable for children. Cure rates are somewhat less than with metronidazole and quinacrine, with side effects like gastrointestinal symptoms, fever, rash, occasionally urticaria, and brown urine. In case of animals, febendazole, also known as Panacur brand name, Drontal Plus brand name, is a preferred choice to treat dogs and cats and found to be completely effective in eliminating experimental Giardia infections with only mild vomiting as probable side effects. Vaccination against Giardia is another option which was shown viable in 1999, where a killed whole organism vaccine was approved for use in dogs in US and found effective to reduce the shedding of Giardia cysts. Prevention and control of Giardia infection can be done through practicing good hygiene, avoiding eating and drinking contaminated stuffs, putting off contact or contamination with feces, etc. Conventional water treatment plants with coagulation, sedimentation, filtration method are needed to prevent waterborne giardiasis flare-ups. Outbreaks are associated with deficiency in community water system with unfiltered chlorinated surface water. The strength of chlorination used for killing most of the pathogenic enteric organisms remains ineffective against Giardia cysts, which require higher concentrations of chlorine and longer contact times to be inactivated. Consumption of hot, properly cooked food helps to avert ingestion of viable cysts from food contaminated by infected water or hands. Giardia virulence. Symptoms or clinical manifestations of giardiasis range from no symptoms to acute or chronic diarrhea, dehydration, abdominal pain, nausea, vomiting, weight loss, weakness, and flatulence. There are many factors like natural immunity, nutrition, intestinal microflora, developmental status, and interaction with co-pathogen, which govern the severity of disease in the host. It is marked that different G. duodenalis assemblages can produce different toxins or metabolic products that contributes to their pathogenicity. However, the results are not very consistent about the relation between G. duodenalis assemblages and virulence, which is primarily indicated by the probability of causing diarrhea and other clinical symptoms, as mostly the symptoms are absent. Researchers have suggested that virulence is more in case of assemblage A. The studies are based from Southeast Asian countries. Assemblage B is often associated with asymptomatic infections. The observations were more evident in children. Serious deviations from the results have also been reported from Netherlands, where association of assemblage A was with irregular diarrhea and that of assemblage B was with duodenal inflammation, nausea, and other important symptoms. Similar results have also been suggested from other parts of the world like Malaysia, Ethiopia, Cuba, etc. The probable reason could be intra-assemblage variation within the assemblages. In a region with an endemic genotype, a slightly different genotype might cause harsh symptoms with its introduction to the population and might also produce synergistic effect in terms of severity of pathogenicity. Factors such as variable surface protein expression, drug resistance, multiplication rate, and ability to invade immune response also contribute to the pathophysiology observed for clinical giardiasis. Socio-economic condition, immunity, and hygiene practices govern the infection rate and other developmental aspects which render virulence of the assemblage A and B in humans. So, student, let us summarize what we have learned in this module. Giardia is a microscopic protozoan parasite which causes giardiasis. Giardia duodenalis can infect human beings. Giardia can survive outside the body for long period of time due to the presence of an outer shell. Giardiasis is the most frequently diagnosed intestinal parasite disease in various countries. Symptoms may vary from no symptom to diarrhea, gas, greasy stool, abdominal cramps, nausea, vomiting, upset stomach, and dehydration. Complex epidemiology of infection can be studied with the help of latest tools and techniques. Diagnosis can be done by demonstration of trophozoites in direct fecal smears, endoscopy to demonstrate the organism in duodenal aspirates, and enzyme-linked immunosorbent assay, also called as ELISA, for Giardia antigen in feces. Giardiasis treatment include antimicrobial drugs such as metronidazole, tinidazole, paromomycin, quinacrine, etc. Giardia duodenalis live in the duodenum and jejunum. Giardia can cause infection through consuming water or ice from unhygienic water sources, ingestion of Giardia accidentally from exterior swallowing water during water sports, especially in lagoon, river, canals, spring, lakes, and ponds, uncooked food containing Giardia, close contact with somebody having Giardia infection or suffering from giardiasis, etc. A considerable amount of data has shown that Giardia duodenalis should be considered as a species complex that comprises at least 8 distinct genetic groups from assemblage A to H. Assemblage A and B, which causes infections to human beings, can also cause infection to other mammals. Humans serve as a key reservoir for G. duodenalis infections in people. It would be premature to comment on the extent of zoonotic nature of infections. The studies have suggested that the rate of infection of G. duodenalis is more in human beings than other animals. There are several methods to diagnose the infections of Giardia in human beings and other animals, like demonstration of trophozoites in fecal smears, fecal flotation, which is a zinc sulfate solution reaction for cyst, collection of duodenal aspirates for the direct demonstration of the organism during endoscopy. Endoscopy is a method in which we use a tube having camera at its interior base. Enzyme-linked immunosorbent assay, also called as ELISA, I have told you already, immunochromatic chromatographic test, and immunofluorescence assay, IFA assay, for Giardia antigen in feces. Factors such as variable surface protein expression, drug resistance, multiplication rate, and ability to invade immune response contribute to the pathophysiology observed for clinical giardiasis. Socioeconomic conditions, immunity, and hygiene practices govern the infection rate and other developmental aspects which render virulence in assemblage A and B in humans. Thank you.